So not only could you have extra chromosomes or missing some chromosomes, it's also possible to have other alterations in the chromosomes themselves. One of the things that could happen is you could have a deletion in a part of the chromosome. So not the whole chromosome is missing in this case. You just might have a piece of it that has broken off and has been lost. Many of them just happen to occur. There's randomness to it. It could also be due to a virus that you were exposed to, radiation, or some other environmental factor that causes that particular part of the chromosome to be deleted. Most of these are lethal or can cause serious disorders because the DNA code is not going to be read right, so it's not going to make the right protein that it should make, and that causes a problem. So you can see in this example here, all you're missing is this C. So the C is missing over in this one, and so it's a shorter deleted portion of it. An example of a deletion is Cridercat syndrome, which is where on the 5th chromosome they're missing a piece of that 5th chromosome, and it causes them to have an abnormal larynx, which makes their sounds that they make sound more like a cat crying, and that's where it gets its name from. It also causes mental retardation in the individual. So another type of alteration that we're going to look at is called a translocation. This is where a piece of a chromosome breaks off and then it reattaches to a non-homologous chromosome. So it's putting genes that don't really belong there. Unlike when we were talking about mitosis and meiosis, we talked about how there's crossing over that occurs, or a piece breaks off and it reattaches to the homologous pair, but it's still just swapping genes. It just might be a different form of that. But this, when it swaps, it's giving a whole new combination of genes that shouldn't be read on that particular chromosome. So the example that we're going to look at is called chronic myelogenous leukemia. We looked at a leukemia before in the cancer unit called acute leukemia. This one is a slower progressor, this particular leukemia, and it's called the Philadelphia chromosome because it was discovered in Philadelphia, and it's caused by a piece of chromosome 22 breaking off and attaching to 9. So 22 is an extra short chromosome instead because it's missing some of that information. It attached to chromosome 9. This causes quite a difference in how that protein is read, and it causes it so that it's not able to regulate the growth of the cell, and instead of being a proto-oncogene, now it becomes an oncogene that can cause lots of complications for that individual. So you can see here that this section right here reattaches to this chromosome right here. So it was on this one right here gains that HIJ, and so it becomes longer while the other one becomes shorter. And this picture over here, these right here are immature white blood cells, and that would be an indication to somebody looking in the microscope at this person's blood that they had the chronic myelogenous leukemia. Right now we'll look at some genetic disorders that occur at the gene level. The issues we were looking at before, remember, they were extra chromosomes or pieces of chromosomes missing, so it involved more than one gene. Now we're going to look at things that affect just the gene itself. So these are inherited conditions based on that alteration in that one particular gene. If it's just a certain gene, it may cause mild symptoms and problems for the person, or it could be very severe depending on what that particular gene that's being mutated. It could cause big problems or just small ones. We're going to start out looking at autosomal recessive disorders, and these are conditions that affect males and females equally. There's no difference in who's more likely to get it. It's all just chance. It usually are children that were the parents were normal, so you wouldn't know that they had this particular gene. They are known as carriers, so they have their genotype is heterozygous for that particular gene. They have one dominant allele and one recessive allele, and it's the recessive allele that if there were 2 copies of it, that's what's going to cause the condition in the individual. So if they're heterozygous, they appear normal and you don't see anything at all in their phenotypes or their genotype. You would just know that if they were tested genetically, you would know they were a carrier. Otherwise, they appear perfectly normal. So this shows you that autosomal recessive inheritance pattern. So if the parents are heterozygous, so you can see this individual has a big A with a little a, and then this one is a big A with a little a as well. And if you do the Punnett square on that, so the eggs are only going to have either a big A or a little a, and the sperm is either going to have a big A or a little a. But when you do the Punnett square of that, 1/4 or 25% of all of those individuals are going to have 2 copies of the recessive trait, and they would be affected by that particular condition. So the most common lethal genetic disease for Caucasians in the U.S. is cystic fibrosis. It's a very common, I'm sure most of you have heard of that. It's caused by a mutation in the CFTR gene, and it affects the endocrine glands, one of the structures we'll learn about a little bit later this semester. But it causes the glands to secrete really thick mucus, and that mucus traps the bacteria. It's harder for them to move that mucus because it is so thick. And so these people are more prone to lung infections, and it requires a lot of treatment and care to make sure that that person doesn't succumb to their lung infections. Another one is Tay-Sachs disease, and this is more common in Jewish, Amish, and French Canadian communities in Quebec. It's caused by a defect in an enzyme that causes accumulation of a sphingolipid in the brain cells, and so it results in the person having neurological degeneration as time progresses, and usually the child will die by the age of 3, so they don't live that long. And as you can see, 11% of the heterozygote frequency in Jewish populations in Eastern European descent. It's more common there. So if you get 2 heterozygotes together, you've got a 1/4 chance in that offspring having Tay-Sachs disease. Another genetic defect is phenylketonuria, or PKU. This one, all newborns are screened for at birth, because as long as that particular genetic defect has been noticed, they can change the diet of the individual and give them diets that are low in phenylalanine. Because they don't have the ability, the mutation in their gene doesn't allow them to convert the phenylalanine to tyrosine, and so they have a buildup of the phenylalanine in their systems, which can lead to brain damage. So as long as they maintain a diet low in phenylalanine, they can go on to lead full lives. So not only are there autosomal recessive disorders, but there can also be autosomal dominant disorders. These two affect males and females equally. It doesn't differentiate between the two. The affected children usually do have affected parents, and also heterozygous individuals will be affected as well. As long as they have one dominant allele, that individual is going to have the condition. Unlike where we looked at cystic fibrosis, you have to have 2 recessive alleles in order to have the condition. So a classic example of this one, the most common one that you would be most likely to have heard of before, is Huntington's disease. And this one causes neurological disorders that leads to the brain degenerating over time because of a toxic byproduct of abnormal proteins, and it destroys the neuron eventually. The problem is that the symptoms don't usually appear until you're between 35 to 45 years old, so the people that are going to be having children don't realize they have Huntington's disease, and they will have already passed it on by the time they start experiencing symptoms, because 35 to 45 is typically after childbearing years. And you can see over here in this chart, the more repeats of CAG that they have, the more the earlier that onset is going to be. So the CAG are those base pairs. So you can see an individual that has 50 repeats of that base pair, the median age to get Huntington's disease is 27, in the range between 24 and 30. But if they only have 39 repeats of those CAGs, the median age is 66. So that ups their chances a little bit of being able to live a fuller life before those symptoms start to appear. And this just shows you, this is pedigree is what this is called. So the squares represent males and the circles represent females. And so this shows a couple, and each of these lines that comes off here. So you can see this individual had 4 children, 3 males and 1 female. And of the offspring, you can see that 2 of them have the condition. So there's a much greater chance of, if it's Huntington's disease, you're going to see more of those in each generation. So a geneticist can put together a pedigree like this, and they can determine how that trait is inherited based on how many they see in each successive generation. And those that have the condition are the filled in circles. Those that do not are the white circles or white squares.